Hello, my name is Hugo Oliveira. I'm a postdoctoral researcher at the University of São Paulo. I'm going to present to you the paper entitled Automatic Segmentation of Posterior Fossa Structures in Pediatric Brain MRIs. Magnetic resonance is a useful tool for assessing the healthy development of infant brains, allowing for the detection of multiple abnormalities, such as tumors, congenital malformations, and hypoplasia. Thus, the segmentation of some areas of interest, such as the posterior fossa structures, is very valuable in clinical settings. However, no public datasets of brain MRIs covering the whole spectrum of infancy, that is, between 0 and 18 years, is available in the literature. There are also no datasets with labeled organs in the posterior fossa region with this age cohort. Therefore, our team decided to create our own dataset in collaboration with radiologists from Instituto da Criança, University of São Paulo. In order to scale the study of posterior fossa structures, we employed machine learning models to conduct the segmentations according to the annotations provided by the physicians. Before presenting our segmentation strategy, we will provide information about our dataset. The 32 volumes acquired by our team are T2 sequences with 4 labeled structures: the fourth ventricle, the brainstem, and the cerebellum. Preprocessing consisted of skull stripping and bias field correction. As you can see in the rightmost part of the slide, most samples are concentrated in lower ages, that is, between 0 and 4 years. 3 samples in our dataset and crops from regions of interest in the posterior fossa structures can be seen here, together with the annotations provided from the specialists. Aiming to improve the segmentation performance, we opted to use an ensemble of segmentation architectures, instead of focusing on one single design philosophy. These networks are the 3D-UNet, the VNet, the HiResNet, the SkipNetNet 3D, and the Med3D network. You can check out all the details of these architectures in our manuscript and in the publicly available source code provided for this research. In our proposed framework, each architecture is first fed a downsampled version of the MRI volume in order to produce an initial coarse segmentation prediction. However, early experiments showed that these initial segmentations were very coarse and tended to produce inconsistent boundaries between classes, even with the use of patching strategies. With this in mind, and in order to account for the large label imbalance in underrepresented classes, we proposed a pipeline with specialist networks. Initially, the generalist network provides a coarse segmentation prediction. This is used to extract 3D crops containing the organs of interest and some padding with surrounding tissue. The first ROI, X1, focuses on the brainstem, in yellow, and the fourth ventricle, in green. The smaller volume is used to train the specialist network, S1. The second ROI, X2, feeds the second specialist network, S2, on segmenting the cerebellum. In the end, a fusion function phi uses the output of the generalist and both specialist networks to provide one single fine-grained prediction. This pipeline is trained for each one of the 5 network architectures individually, and their predictions are merged using majority voting. In order to properly evaluate the performance of the generalist and specialist settings, as well as the majority voting, 3 distinct quantitative evaluation metrics were employed. These are the voxel-wise dice score, 2 variations of surface distance metrics, and the R2 correlations between predicted and manually annotated volumetries. These 2 tables summarize the results of voxel-wise dice and the surface distance metrics for our specialist pipeline and a generalist pipeline as a baseline. We also include individual segmentation architectures for the dice metric, in comparison with the majority voting. As you may see in the highlighted results, both the majority voting and the use of specialist networks consistently improve the performance of the algorithm on dice and surface distances. The correlations between manual annotations on the y-axis and the network predictions on the x-axis also improved by a large margin when employing the specialists. The best R2 for the brainstem reached 0.83, while the cerebellum volumetry from the specialist network and the radiologists were highly correlated, reaching 0.94. At last, we present some qualitative results for the same volumes shown previously on this presentation. Relying simply on the generalist networks yields very pixelated boundaries between classes. When using only the S1 specialist, the segmentation of the brainstem and the fourth ventricle is clearly improved, but there are inconsistencies in the boundary with the cerebellum. All of the aforementioned problems are considerably mitigated when using both specialist networks, wherein the boundaries are clearly delineated and very close to the ground truth annotations. As our final remarks, we observed that the agreement between ground truths and predictions was high even in a very small data scenario with only 32 samples. Also, the use of specialist networks and majority voting proved to be a highly successful strategy for the segmentation of posterior fossa structures. At last, given the accuracy of the volumetry predictions, our pipeline could be used to define volumetry standards for healthy pediatric neurological development. Future works include the analysis of developmental abnormalities in our dataset, gathering additional unlabeled data from Instituto da Criança for the creation of the volumetry standards, and testing our pipeline on the recently published FETA challenge. Our team is composed of graduate students, postdoctoral researchers, professors, and radiologists. On the rightmost side of the screen, you can access the GitHub for this project via the QR code or the link. The authors would like to acknowledge the financial support provided by Fundação de Apoio à Pesquisa do Estado de São Paulo and Agência Nacional da Recherche.